Hi everyone, today we're going to look at math and genetics, and we're going to look at probability and chi-square analysis in genetics problems. So a little bit of review before we get started. You should remember that we have chromosomes. On those chromosomes are genes, and for each gene we can have different alleles. An allele is just a version. So here we have a gene with the allele for purple flowers, and here we have a gene with the allele for white flowers. We know that there are dominant and recessive genes. If we have two alleles that are the same, dominant or recessive, we call those homozygous. If we have two that are different, we call it heterozygous. We know that the phenotype is the physical expression of the trait, whereas the genotype is the genetic makeup. And we've done Punnett squares. Okay, that's our tool for predicting our ratios. We've done genotypic ratios and phenotypic ratios. And that's where we get to start our math. So we talked about segregation of alleles and fertilization as chance events. We talked about that in the meiosis and mitosis videos. So we know that it's like a 50-50 chance as to whether or not the sperm gets the capital or the lowercase. Now, this is if the parent is heterozygous. So it could be the capital R or the lowercase. Same thing for the mother who is heterozygous. Okay. Once we move our information together, our chances of having a capital R, in this case 2 heads, is 1 out of 4, which is 25%. Our chances of getting a capital and a lowercase, or a lowercase and a capital, because we don't care about the order, that's 2 out of 4, or 50%. And the chances of us getting 2 lowercase, 2 tails, that is 1 out of 4 boxes. So that's 25%. Now I can look at this further and say, let's say we flipped this penny 100 times. I would expect out of those 100 times that I'd get an equal number in each box. So I would take 100, divide it by 4, and each box would get 25. That means that if I flipped a coin 100 times, I would expect to get 25 that are this way, 25 here, 25 here, 25 here. If I did, it's a set of 2. So if I flipped those, I would expect to get 25 in each. That means I would expect to get 25 sets that are both heads, 25 sets that are head, tail. Oh, wait, 25 more that are tail, head. So that's 50 that are one of each. And then 25 that are 2 tails. Okay, so again, if I flipped coins 100 times, so a set of 2 coins 100 times, looking to see what results I would get, I would expect to get 25 of the coins like this, 25 like this, 25 like this, and 25 like this. That means I would expect 25 coins to be heads, heads, 50 to have one of each, and 25 to have tails, tails. Now we can look at what is called the law of multiplication in probability. Now for multiplication, if we say, what are the chances of this and this happening at the same time? Okay, then I take the chances of one and the chances of the other and multiply it together. If we were to look at dice, the probability of throwing 2 sixes. If I threw 2 dice, the chance of getting a 6 is 1 out of 6 on the first dice and 1 out of 6 on the second dice. Because I want to know what the chances of getting them together, and I'm going to multiply. So 1/6 times 1/6 is 1 out of 36. Okay, the probability of having 5 boys in a row if you're having kids, well, it's a 50-50 chance the first time, but it's also 50-50 the next time, and the next, and the next, and the next. And I want to know, what are the chances of having boy and boy and boy and boy and boy? Okay, when I do that multiplication, I get 1 out of 32. All right, so here's a problem here. What is the probability of having 2 boys and 1 girl? Okay, 2 boys and 1 girl. Well, the chance of having a boy is 1/2. The chance of having another boy is 1/2. The chance of having a girl is 1/2. So I just multiply out. Now this next one is a little trickier, and it's kind of important to be able to do these. So I'm going to move to my camera so that we can do this one together. There we go. If I cross a parent that is capital A, capital A, capital B, lowercase b, capital C, lowercase c. So homozygous dominant, heterozygous, heterozygous, with one that is heterozygous for all traits. Oh, move that over a little so you can see. There we go. Okay, what is the probability of having offspring that is, let's see, this. Now, in order to do this, we can actually do just 3 small Punnett squares. Okay, if I do my A's, then do my B's, then I do my C's. Now you may have noticed that I put a little mark on my capital C. That's so when I'm writing in here, I don't get them confused. What's a capital and the lowercase? I know that the one with the little mark on it is always my capital. Okay, so then I look at this one. What are the chances of getting a heterozygote? Well, it's 2 out of 4 or 0.5. And then if I look at my B's, the chance of getting the heterozygote there is going to be 0.5. And then if I look at my C's, the chance of getting homozygous recessive, okay, that one is just this one box, so that's 0.25. So if we do that math, grab my calculator here. All right, 0.5 times 0.5 times 0.25. That equals 0.0625. Now I can change that to a percentage by moving my decimal 2 places, and I get 6.25% chance that they have offspring that looks just like this. That's much better than doing giant boxes with 3 gametes for each box and then putting them together. So not so bad, right? Now let's go back to our presentation. Okay, next we have the rule of addition. This is saying the probability of something happening if we want this or that. So if we have one die and we throw it and we want to know what are the chances that we could get a 4 or a 5, then we add. So a 4 would be 1 out of 6 and a 5 would be 1 out of 6. When I add 1/6 plus 1/6, I get 1/3. So it's important to remember that multiplication is and, addition is or. Okay, chi-squared analysis. Biologists and other scientists use relationships that they've discovered in the lab to predict events that might happen under more real-life circumstances. At some point, the predictions are either supported or not by real data. But it would be nice if two sets of data matched exactly, but more often than not, they don't. So in the instance when we talked about having 100 sets of 2 coin flips and looking at what our results were, we would expect to get 25 that are heads. Okay? But that might not happen. We might not get exactly 25, 50, and 25. So chi-square analysis lets us test to see if that is because of random chance or if something more is going on. For instance, if I was a casino and at a craps table with dice, if we got things that were out of whack, we might want to know, are those dice loaded? Is everybody playing fair? The other thing is we can use this in nature. So if we had 2 trees and ants in the area, we would expect half of the ants would probably be near one tree and half near the other. And if we did this analysis, it could show us that the ants actually favor one tree over the other, and that could be because of sap type. It could be because of aeration in the soil. It could be because of the amount of sunlight that filters through. Lots of different things, but it tells us if we need to look further. Okay, so we're looking to see, is it random fluctuations or is there something else going on? This calculation is called chi-square. Now you'll notice that it's in italics X with the square. That is where we leave it. We do not square root any of these things. So x squared, that is chi-square, and that's the number we want. And then it has all this craziness over here, which everybody freaks out about, but it's not a big deal. You will get all of this information on your formula sheet for the AP exam. Okay, so here is chi-square. Chi-square equals sigma O minus E squared over E. Now, these could be capitals or lowercase. It doesn't matter. It means the same thing. O is our observed, what we actually see in real life. E is what we would expect it to be. And sigma, this big funky looking E kind of letter, sigma just means do it for all of them and add them up. And then another thing that's important is degrees of freedom. So degrees of freedom are the number of distinct possible outcomes minus 1. Okay, so let's take a little bit more closer look. This is more information that's, you're going to have to use it to understand it really. But we're going to take whatever that number comes out to be, and we're going to compare it to this table. And we have, you can see degrees of freedom. Okay, if we were looking at colors of Skittles, okay, if there are 5 colors of Skittles, then our degrees of freedom would be 4. And we could count how many of each we have in a package and see, does it match what they say they put in there? Okay, so degrees of freedom are whatever our possibilities are minus 1. And then we have our p-value. Now the p-value, this is our probability. It tells us what kind of confidence we're talking about. And we're always going to use 0.05. This says that we are 95% sure that we're giving you the right information. So if we were looking at Skittles with 5 colors in them, we would compare the number we got from that calculation to this number. So 5 minus 1 is 4, and a p-value of 0.05. Where they come together is 9.49. And when we compare them, that gives us a lot of information. Okay, so let's look at how this works. First, we have to be able to create a null hypothesis. Okay, our null hypothesis is saying that there's no statistical difference between what's happening and what we would expect. Okay. So if I were to flip a coin 100 times, I would expect 50 heads and 50 tails. And my null hypothesis would be that the expected outcome is to have 50 heads and 50 tails, and any variation is simply due to experimental error. Okay, so that's our null hypothesis. It means that there's nothing wrong. Everything's happening how it's supposed to. We think things are fine. That's our null hypothesis. Okay, so let's take a look here. A poker dealing machine is supposed to deal cards at random as if it has an infinite deck. in a test, you counted 1,600 cards and observed the following. Okay, now we can make a table for chi-square. So we're going to check out that table. We're going to fill it in using this information. So let's go ahead and go to the other camera. All right, so first thing we have to do is think about our null hypothesis. Now, our null hypothesis would be saying that there's nothing wrong, everything's happening how we expected. Now, we have 1,600 cards, 1,600 cards. And if they are dealt, now for those of you who don't know, playing cards have 4 suits, they call them. Spades, hearts, diamonds, and clubs. Now, the spades are the ones that kind of look like that pointy shovel. Yeah. Okay, so when we look at our information, the first thing we have to do is develop a null hypothesis. So our null hypothesis in this case is going to be that everything is as we would expect it to be. So if I have 4 different suits and 1,600 cards, just like when I had my 100 flips and I put them in the 4 boxes, I have 4 boxes and 1,600 cards. So I would expect them to be divided evenly, meaning I would expect 400, 400, 400, and 400. So our null hypothesis would be that each suit has 400, whoa, not 4,000, 400, has 400 cards. And any variation is due to sampling. Now, it tells me on the slide that we were just looking at that for spades, we had 404. For hearts, we had 420. For diamonds, we had 400. And for clubs, we had 376. So that's our observed. Now, those are pretty close to 400, but should we trust them? So our first step is to take our observed and our expected. We subtract expected from our observed. So in here, we're going to have 4, 20, 0, and negative 24. Now, negative numbers are fine. It's okay. Because what we're going to do next is square this. When I square things, I get rid of those negative numbers. So this is going to be 16. Grab my calculator, make sure I don't do these wrong. So 20 squared is 400. 0 squared is 0. And negative 24 times negative 24, 576. That's a lot. Okay. Now, we take these and we divide by the expected. Okay? Move it over a little bit so we can see, okay? It happened to be that this time we expected them to be equal 400 for each. So we know that this is going to be 0 and this one's going to be 1. If I take 16 and I divide that by 400, I get 0.04. Now if I take 576 and divide that by 400, I get 1.44. Now this seems like it's an awfully big number, pretty far away, but we're not done. We have this information. Now we're going to add those up. Okay, so this gives me 1.48. 1.48. So our chi-squared sum is 1.48. Now, our degrees of freedom. Okay, next is degrees of freedom. We had 4 different suits: spades, hearts, diamonds, and clubs. So 4 possible outcomes. I subtract 1. That means my degrees of freedom are 3. Now I need that table. Let me find my table real quick. So here we go. Here's our chi-squared table. I know that my degrees of freedom were 3, so that's right here. I'm still going to use my confidence of 0.05. That means the number I'm going to compare it to is 7.82. Make sure that's focused there. There we go. All right, so we want to compare this number to 7.82. Now, our number was 1.48. Okay. And then our number we're comparing it to is 7.82. That means we are lower than our chi-squared value. Okay, lower. If we are lower than the chi-squared value, we accept our null hypothesis. Okay, so we are going to, now, saying that it's right, we're actually going to say that we fail to reject. This is our proper terminology. We can either reject the null hypothesis or fail to reject the null hypothesis. So we are going to fail to reject our null hypothesis, and we are going to say that there are 400 of each suit, and any variation is due to random, chance, or error. Now, if that number had turned out to be larger, where's my table? There we go. Let's say that number had come out to be 10.48. Well, that would be larger than our chi-squared value. Than our sample value. And that means that we would reject our null hypothesis. Okay, that means we could say, no, something's wrong here. We need to figure out what's going on. Okay, so we have one more example we're going to try. So it has been observed that eye color in fruit flies comes in wild type and bright red. Wild type is dominant. In a cross between a female with wild type and male with wild type, the following was observed. Okay, so we know that wild type is dominant. First thing we need to do is we can pick a letter. So let's use E for eyes. And E is going to be wild, the capital E. Our lowercase e is going to be bright red. Wild type red, by the way, is kind of a darker red. So there are 2 reds, one bright, one dark. Okay. It says in a cross between a female with wild type. So I know there's at least one of those. And a male with wild type, at least one of those. The following was observed. 500 offspring. So I have 500, and I know that 380 have at least one of these, and 120 have that. Well, the first thing that I know is that these have 2 little e's. They are homozygous recessive. That means they had to get one from each parent. So I'm able to fill that in. Okay, the next thing I can do is I can see that this was a basic monohybrid cross. Okay, in a monohybrid cross, if I sketched that really quickly, I know that I had 120 that were in this area and 380 went in the other 3 areas. So if I were to divide those up, okay, I'd have, say 127 roughly in each of those. Okay, so let's see what the questions are asking. What would be the null hypothesis if you were to test this cross using chi-squared analysis? Okay, so if we were to take 500, so we had 500 total. If I had 500 that went into here, then wouldn't I expect to have 125 that are homozygous dominant, 250 that are heterozygous, and 125 that are homozygous recessive? That means that I would expect to have 375 wild and 125 red. So this is what I would expect. So my null hypothesis is going to say that with 500 offspring of this cross, we would expect to see 375 wild and 125 red. Now, what is the chi value and what does it tell you? So we can easily perform our chi-squared analysis on this. Let me just give us some space here to work. We had wild and red. We need our observed, our expected, observed minus expected, expected squared, observed minus expected, oops, squared divided by expected. So these are the numbers that we need. And then we're going to add them all up. Okay, so again, you'll be given all the information you need on your formula sheet. So you'll have this on the formula sheet. So the information you need is what is O? What is E? What is O minus E? What is O minus E squared? Okay, then you can figure everything out. So our expected wild type is 375 and 125. What we actually found was 380 and 120. The difference here is 5 and negative 5. Squared is 25, 25. Okay, now notice that our expected is not the same this time. Okay, what is expected is going to depend on what your formula is, what your situation is, what your problem is. So in this case, I'm going to take my observed of minus expected squared of 25, so 25, and I'm going to divide that by my expected 375. And I get 0.0. Now we're always going to round to 2 decimal places unless it tells us not to. but we want to do that at the very end. Okay, we want to do that at the end. So if you can leave it in your calculator, do. Otherwise, I usually go out about 4. Now we're going to take 25, and this time we divide it by this expected, which is 125. And that gives us 0.2. Okay, so if we add them together, we get 0.27. If I take a look back at my chi-squared table, I had 2 outcomes. I had red or wild type. So because I have 2 outcomes, my degrees of freedom, okay, so degrees of freedom are how many? -1. 2 minus 1 is 1. So 1, okay, and we're looking at p equals 0.05. So the critical value is 3.84. So our critical value equals 3.84. And our chi-square value equals 0.27. Okay, so that means we are less than the critical value. If we're less than the critical value, we fail to reject our null hypothesis. Okay, so we are going to fail to reject. This means that our null hypothesis saying that there would be 375 wild type and 125 red, and that any difference would be simply due to either error or random chance. Okay, that is correct.